let me start with the HIBALL closing symposium. And it's difficult to start and not to remember where we started, practically. We started long before the HIBALL Joint Lab has been organized. It began in the middle or beginning of the 2005, 2013. And the reason why it took so long was, at that time, we decided with Carl Zillis, with Alan Evans, to process an entire human brain, section by section, and to 3D reconstruct it. And when we started, it was absolutely not clear, and there was no good way to reconstruct this histological section. Nobody has ever done such an endeavor before. And it was challenging both from the lab point of view, how to process section, how to stain them, how to digitize them, how to count them in the right way, how to make a quality check, and then how to reconstruct these sections. And it took more than 8 years. We started in Düsseldorf at that time, but then I would say we were lucky enough to place this joint paper, very high in science. And after that, we really tried to develop the big brain into a resource of the community. So not just have a cool reconstruction, which is of course breathtaking when you think about the detail, but really provide this as a resource to the community and enable other type of research. And Ellen and me had in mind that this research should be organized around several key topics, and they are reflected in the work packages. And when we organized this joint lab, we took care that all these work packages, they have joint contributions. Not always 50-50, I mean, science is not like this, but in a balanced and in a, so to say, in a way that allowed to make the strengths and the expertises in the different labs visible and created some synergy. And this is the first board of work packages, and it's really a shame to say it, but when we started it, we overlooked a little bit the management and administration. And when you are today here and you see how everything is well organized, how people can speak with each other, then it's of course PJ's and Susanne's merit very much, who are the last work package. I would say also one of the key and central work packages, because it allows us really to keep together, to work together. So if you allow me already at the beginning to say thanks to both and their little teams, and please join me to thank them. So we also had the goal from the very beginning to support younger researchers, and these are only a few of them. So from the very beginning, we understood that it's important that younger researchers, PhD students, get the opportunity to travel, to learn new methods, and to work in the other labs. And these are some of them. And some of them really had an impressive career meanwhile, and profited and benefited, of course, from this collaboration. And we are, of course, particularly proud of those. And Hannah is here, Jordan is here, and Christian is also here, and many of others are also here who traveled back and forth. Then we also had very nice events in Montreal here, where the fourth BigBrain Workshop was, and Montreal really has a separate lab space, where HIBALL is visible. And this seems to be, for some, maybe a minor thing, but you need to have a place also, where you can show this is HIBALL. I mean, it's not just something virtual hanging in the air. It's really very concrete. There is cool stuff. There are computers. There are displays. And I remember that Ellen had really one of the very large first screen vaults when it was not so popular and also much more expensive than it is today. But it was important because we wanted to go into the details of the cytoarchitecture of the big brain. And this place was then also attractive at the political level, and our former minister, Bettina Schwarz-Watzinger, Bettina? Sorry. Okay, so she visited Montreal, and also the chancellor was, at that time, Olaf Scholz, visited Montreal and the Mila. And this high visibility was, of course, also important for the support of the HIBALL initiative. And there was a lot to show at such occasions. And for example, HIBALL provided and is providing tools to the community that everybody can use, and that are distributed through our website and other websites like the Zebra Explorer or the MicroDraw, NeuroWeb's Brain Box, Brain Browser. So these are all dedicated tools that have been developed to enable better brain research by collaborators and by parts, by members of the HIBALL consortium. And along the same line of reasoning, we are also providing maps and models to the community, and also provided as a free resource that can be downloaded and that can be implemented in different other workflows, and you will hear about it a bit later during this day. And there are many very high-level papers published, and this is emphasized because when we are able to have publications in such journals as listed here, this has, of course, also a different way to be visible. It's higher visible. And then other researchers can benefit from what we are doing, and they benefited. And just let me give you some numbers, BigBrainProject in numbers. You see here that there are 90,000 downloads from the FTP side of the BigBrain, 24,000 visits at the eBrain Zebra Explorer, which is closely related. Lots of views on YouTube videos and new visitors, 170 publications. I think that's quite a lot in 5 years, yeah, for such a dedicated project, which is not only about scientific application, but which is very much, of course, about methods development, infrastructure development. And all of you who are programming, who are doing such work, they know how difficult it is and how long does it take to develop it. So we did not start from scratch, but 20 years ago, as I illustrated, so we have a trust, we have a way to collaborate. I think this was extremely important for the success. So now let me give you some ideas what HIBALL science means, and these are a few examples of many. And let me start with Hannah Spitzer, who has been a doctoral student in Jülich, working with Timo Dickscheid, and now she has her own research group in Munich. So Hannah is also here. So she started to use deep learning for brain mapping. And this was really a painful process. The first maps looked quite nice, I would say, for the primary visual cortex, but not so great for other parts. And then step by step, we were able to further develop the method and use deep learning for brain mapping. And the results became more and more accurate. And CytoNet, that is introduced today by Christian in more detail, has, of course, this tradition and is based on what Hannah did a couple of years ago. So we are now able to much more precisely not only identify the localization of areas, but also to classify areas. That's a big difference from the classical mapping approaches. So we know it is area X or Y. And these new models, CytoNet allows also to segment the layers in the big brain to quantify generative models, doing data-driven discovery of brain structure, and predict interpretable features. And I would say it was not clear when we started that AI is making such a jump and such a dynamic development, but it turned out that really what we planned 6-7 years ago, I would say, came true, and the developments that we see in the AI field are really impressive, and we are, so to say, we are pioneering this type of research. So there will be talk today by Christian a little bit later. We also added during the years more and more maps to the big brain in terms of cortical areas. And we had to do quite some effort because the big brain is one of the newer brains of the Jülich collection of post-mortem brains, and we did not use it at the very beginning for mapping and creating probabilistic maps. So the big brain came later, and we then had to solve the question how to make it comparable. And during the last few months, thanks to Kimberly Lodman, we have really a lot of new areas mapped, 56 new areas. We will make them available. There's a poster, you will see it immediately, and please inform yourself what is going on. One of the structures that we mapped was also the hippocampus. And the hippocampus can be mapped, of course, at different levels of spatial resolution, and it should be mapped at different levels of spatial resolution, because when we think about patients and MR images, then we want to make these maps applicable to MR images. And Jordan de Kraka, also one of our postdoctoral students, has just recently published a very nice paper showing a method to deal with the hippocampus. Why it is so difficult? Well, the hippocampus has not only a very difficult shape in the sense of the hippocampus, but there are also bumps which are very specific to every individual brain. And we can map the brain at the same time also in the big brain with ultra high resolution, where we see every and each single detail, and then we can inform each other. Yeah? So postmortem can inform MR imaging and vice versa. So I think that's a big progress. Another result of the last time is the so-called BigBrain 2. So this BigBrain 2 is also a brain of a body donor, and it is now reconstructed at 100 micrometer. Claude Lepage and Hartmut Mohlberg are the two guys who take care, supported by Lindsay Lewis and by others in the lab. And the reason why we started the second big brain was to get a bit more understanding of what is variation, what is variability. But also the second big brain is better just from a histological point of view. It has less artifacts. Yeah? So also this we had to learn how to process, and we think that that will also be an important resource to the community, and we will further increase, of course, the spatial resolution. Because at the end, I mean, we want to go to the cellular level. Yeah? The big brain has the vision behind that we want to understand the cells in the human brain, and there are many of them. There are 86 billion. And of course, it's not possible to just classify these cells based on traditional image analysis. I mean, people did it forever, I would say. And only when deep learning has been developed and Erik Upschulte and team started to use, then it was possible also to solve this question. And we have now a method available that allows to segment cells, like you can see here, and that solves also the problem of cells when they are stitching, when they are a little bit overlapping. And to distinguish these cells is extremely important when we do any statistics, when we want to quantify them. And the huge amount of cells, I think, becomes clear when we simply zoom out now and go to the level of the cerebral cortex, as you can see here, or when we go further to whole brain sections. So this is an extremely time-consuming and labor-intensive, in particular at the beginning, endeavor, but it helps us then also to do the next step and to 3D reconstruct at this level. So cell numbers are, of course, a goal, and we do not want only to replicate the 86 billion nerve cells from the literature, but to be more precise, when I'm in a certain cortical area, in a certain cortical layer, what is the cell density and what is the absolute cell numbers? And when you think about simulating the brain in healthy subjects, or when you think about modeling and simulation also to prepare surgery, for example, in epilepsy, then such numbers are extremely important. But for the human brain, they simply did not exist until now. Yeah? For the macaque brain, for the mouse brain, yes, but not for the human brain. So what we are doing now, we are quantifying big brain cortical densities in many different regions in the brain and also in different brain areas, and we quantify them, showing what is the distribution of cells from the surface to the cortex white matter, independently on how the brain is cut and processed. From the very beginning, one big challenge was that during histology, during cutting, staining, mounting, always artifacts appear. They are inevitable when you think about 7,000 histological sections. And then the question is, what are we doing with these artifacts? Yeah? Again, there are a lot of them, and just to handle them and paint them one by one is simply not possible. It does not scale up. So Oliver Kropp has developed methods to help to repair such artifacts, and it looks a little bit like mystery, I would say, when you watch this video. So there is no tissue, then there is some tissue. It looks quite okay, I would say. Is this enough that it looks quite okay? I would say not really, yeah? Because the next step is then what has been repainted. Does it reflect the true features of the tissue? And Oliver Kropp did some analysis and reality check and asked, is the classification consistent with what we see from a neighboring region? Are the cell statistics consistent? So it's not just predicting and then we take it. It's really having this productive loop and indicating this is a true number. So we have some provenance tracking. We know where we have prepared and repaired, but we also know that the statistics of this repaired tissue are the correct one. And this is really a major, so to say, this is a major step for human brain tissue in particular, which is so huge and so to say, so easy to damage. So again, there's a talk. Please visit. And cytoarchitecture is a very good starting point, and this is a case because there are many, many other types of data that relate to cytoarchitecture about connectivity, function. There's a lot of information on it, so it's like a reference modality, not the only one, but a very convenient one. But at the end, we want to understand how the cytoarchitecture is related to the axonal architecture. And in this image here from Roxana Koyman, we also see immunostaining of PAF-albumin, calbindin, and calretinine positive neurons. So it's a combination of the big brain approach, but with a specific classification of cell types and making visible the axonal architecture. And the axonal architecture can be made visible by polarized light imaging that is developed in Markus Axer's lab. And he and Boris, Boris Bernhard were leading work package 2. And we want to bring it together, of course, also with a molecular architecture, with a receptor architecture, which is so important for signal transduction and makes the human brain so different from any artificial system, from any in silico system. Now, we have chemical signal transduction. That's quite a lot of possibilities to modulate signals and to change information processing in the human brain. And of course, we want to understand what are the bases, and we want also to make it helpful for better understanding how drugs are, for example, binding. But when you think about so many modalities, then it is also clear that you have to find a way to combine all these different modalities. And Alexander Oberstraß will give a talk later today and introduce Tiamat for tiled image access, manipulation, and analysis toolkit. And you see all kinds of modality that he will address. So let me, at the end of my talk, give you a little bit an outlook of the clinical application that's coming from the big brain and coming from anatomy. And one of the most frequently used targets of deep brain stimulation of patients with Parkinson is the so-called nucleus ventralis intermedius, or VIM. And this VIM is a structure of the thalamus, and it has been mapped by Hassler, who was a Frankfurt neuroscientist, and then became part of the Schaltenbrand-Waren Atlas that is used in surgery for localizing this VIM. But when you have a look to another nomenclature of Jones and Morel, and this is very much used in the neuroscience field, very much also, I would say, supported by the American school, then there is no VIM. Simply this nucleus is not mapped, and the argument is that it is part of another nucleus of the thalamus. So the question was, of course, does it exist? And that's not only an academic question, I would say, between different nomenclatures. So we started with a doctoral student to map the VIM in histological sections based on the Schaltenbrand-Waren Atlas and the description. And on the right-hand side, you see three zoomed images of three nuclei that are located, the VLP, the VIM then, and the VPL. And it was rather easy to identify the VIM. I mean, not as difficult as I thought, because it has these really prominent dark, large cells, which is also typical for some motor areas. So we have identified this nucleus. And then the question was, is this really a subnucleus of VLP or VPL? And the way we discussed it was, or we solved this question was, we did some texture analysis and compared the texture of these three nuclei. And the hypothesis was, if it is a subnucleus, then there should be a higher similarity to one of the larger nuclei. But this was not the case. So all three nuclei differ to the same degree between each other, which provides a cytoarchitectonic argument that this is really an independent nucleus and not just part of something else. And we mapped now this nucleus in 10 brains and compared it here with a post-operative CT of a patient undergoing surgery. And you see here that it could help also to better understand where's the localization of electrodes and maybe also to follow up. But this is not my field, and I'm really happy that we were able to get Andreas Horn for a keynote tomorrow. So he is working precisely in this field and using the big brain as a template, as a high-resolution template. And we are very much looking forward to this talk tomorrow. So these were only some examples, and it took already quite some time. But now we have a second round, I would say, of really impressive examples from science. And I would like to hand over to Professor Ellen Evans, my dear friend and colleague over the time, with whom they built that. And you have mentioned already that sometimes it is difficult to distinguish. Will you show it? Will I show it? I think that's not so important at the end. But now please show your examples.
Thank you very much, Katrin. So this is a strange meeting in some ways because it's called the closing symposium, and I don't see any sign of HIBALL closing scientifically. It started, Katrin and I have been working for about 20 years on started out at the BigBrain Project, and that grew into HIBALL. On our side, yesterday, some of you were present when we had the HBHL, Healthy Brains, Healthy Lives, training day. We had a wonderful lecture from Helen Zhao, talked about big brain, large-scale big data, artificial intelligence, and neuroscience. And then we talked a little bit about HBHL activities. It's a sprawling enterprise HBHL of over 200 PIs. That gave rise to HIBALL 7 years ago on our side with funding from the HBHL. On the German side, those fundings were matched by the Helmholtz Association, and that gave rise to HIBALL. And I have to say that I've been involved in many collaborations over the years, and there's no question in my mind that the HIBALL collaboration with Katrin and Katrin's group has been an ideal organizational exercise. Lots of cross-fertilization scientifically, exchange of students, exchange of ideas. It's been a wonderful experience, and this is not fin de siècle. We've just come to the end of the first chapter. We'll be talking more about the future at the end of the day. It falls to me to just make a few comments on further scientific advances. As Katrin pointed out, there are 170 publications that come directly from the HIBALL initiative. That's only those which have come directly. There are a further 200 publications which have come from groups using HIBALL products. So if you look at it from that perspective, you're approaching 350 or more publications that are HIBALL related over the last 5 years. So I'll just show you just a couple of highlights of some of the other projects that we've been involved in and continue to be involved in within the HIBALL community. One of our earliest HIBALL products was the work of Konrad Wagstill and Hanna Spitzer, who developed a fully automated machine learning, deep learning approach to the segmentation of the 6 cortical layers. They used a one-dimensional convolutional neural network approach to identify the 6 layers. And it's worth bearing in mind, this was fully automated in both hemispheres over the whole brain. And this is to be compared with the traditional work that we're all familiar with from von Economo and Brodmann in the early 1900s, where they identified brain areas and brain layers, often by visual inspection. All of this was done in a fully automated, reproducible manner. And when you compare the results of this automated approach to von Economo, for instance, you see on that graph in the middle there, a comparison of the big brain cortical thickness versus von Economo. Very good correlation between the two, in both hemispheres. Similarly, when you compare those layer thicknesses from the Human Connectome project from MRI data, again, very good correspondence between the big brain automated layer analysis and those obtained from the HCP. On the bottom there, you see the 6 individual layers across both hemispheres. Another project that we've been involved in for some time, I always like this video. I can stare at it for hours. Cerebellum is one of my favorite organ systems in the brain. You see here work from Xuan Liu And this is the fully automated segmentation of the big brain cerebellum. On that video, you can see the 6 cortical layers in the cerebrum, and that's just, by the way, the main focus of this video is the segmentation of the cerebellum. As I say, it's really quite hypnotic. I'm going to let it run for just a few more seconds because I like looking at it. Okay. More recently, Navona Kalako and Camille Uladag have taken the big brain and automatically segmented the colostrum. Now, we've all heard the commentaries that the colostrum is somehow associated with consciousness, and we'll let that one float in the air for some time. What we didn't know is that the colostrum is a really extensive structure. Most of us used to look at the colostrum as that thin stretch between the caudate nucleus and the thalamus. And when you look at it in the big brain, you see that it's a very large sheet structure. And this is a comparison of the colostrum as derived from the big brain by Navona, and a comparison with the 7T MRI structure. And there's a pretty good correspondence. As you see on the left, this is when you're looking at compared with the big brain derived gold standard versus MRI. And on the right, you have, after we've downsampled the big brain to make it more comparable with the MRI. And there's a pretty good correspondence. Now, Katrin mentioned this one, and as always, we didn't coordinate our talks entirely perfectly. Jordan, who's in the room, Jordan de Cracker, did his PhD work at Western University with Ali Khan, and then came to Montreal, was working with Boris and myself to work on the 3D reconstruction of the hippocampus. And as you can see on the right, there's the hippocampal segmentation of the big brain. Something that I was completely unaware of before Jordan did this work is that the hippocampus has sulci, as pointed out on the bottom right of the slide. It's still an area that we haven't begun to do, and we really should explore the relationship between those hippocampal sulci and behavior across individual populations. We can use this kind of information to inform the segmentation of high-field MRI of the hippocampus, but that's for the future. So what you see here with Jordan's work is the subfield segmentation. You can then take those subfields and map those from a 3D native coordinate space onto a 2D coordinate space, the eponymous hip unfold procedure that Jordan has developed and promulgated quite extensively. Lots of people around the world are now using the hip unfold machinery to compare across individual subjects within a common two-dimensional hippocampal coordinate frame. Hip Unfold is available to anybody out there who wants to use it through the CBrain platform. It's one of the many work packages, workflows that are made available through CBrain. You heard a lot about this yesterday from Brian Cadon, and there's another talk today which goes into further detail about the uses of CBrain to support extensive analytic packages. HippoMaps is a further extension from Jordan, which uses that two-dimensional coordinate framework as the basis to make an open repository of standardized maps of histology, structural MRI, functional MRI, intracranial EEG. So lots of data can be composited within this common coordinate frame. Moving on, those of you who were here yesterday had the pleasure of listening to Boris Bernhard summarize what's going on in his laboratory. And I'm constantly amazed at how many papers and PhD projects that Boris manages to coordinate simultaneously. A few examples are shown here, the Mica pipe analytic workflow, The HIP Unfold that I just talked about is now made available through Boris's Mika lab. And more recently, Yungan Huang has developed a superficial white matter analysis package to explore the subtleties of the superficial white matter near the sulcal boundaries. Those are the analytic methods. You can look at also data warehouses. We've already talked about the HippoMaps data warehouse, but there's a new precision neuroimaging and connectomics database that was made available. Boris talked about this yesterday. And this involves now 30 subjects, 3 sessions, an extensive 7 Tesla MRI protocol. As you can see down below, many different MRI protocols, as well as episodic memory and retrieval data that's shown at the bottom right there. Boris's lab, particularly people like Casey Piccola in the past, have shown leadership in recharacterizing brain organization and brain mapping through a gradient approach rather than a regional approach. And one can talk for a long time about the evolution of the brain through a gradient perspective rather than through a regional perspective. Moving on to 3D chemoarchitecture. You've all seen over many years Carl Zillis's original autoradiography data of 20 different receptors. This is obviously work that came from Mulish originally. And although Carl dined out for a very long time on these gorgeous autoradiography images, they were in 2 dimensions. Thomas Funk in Montreal at the time put in the efforts to allow us to take those 2-dimensional slices, often separated by large distances across the brain, and interpolate those into fully 3-dimensional maps. You see in this particular image on the left, a 3-dimensional GABA-benzodiazepine map. Here's the challenges that were faced. Thomas is not the challenge, it's the brain problems are the challenge. And Thomas spent a good portion of his PhD in Montreal working on the mechanical problems of nonlinear deformation, variability in pixel intensity, large separation across sections in brain, and variability in digitization. And that has been published, but now very soon you will start to see not just the methodology of this 3-dimensional autoradiography, but the 20 different neuroreceptor maps will be coming out repeatedly for each of those individual receptors. Thomas has also gone on to apply the same procedures that he developed in the human context, now in the macaque brain, to look at cell body myelin density and binding site density in the macaque model. Now this is all autoradiography. Justine Hansen, with the supervisor, Bratislav Mizik, came at this approach from a different direction. They took in vivo PET receptor maps from 26 different sources, and they built up 19 receptor and transporter systems and 9 transmitter systems from the brain. This was published back in 2022, and you can see these beautiful three-dimensional PET maps of neuroreceptor neurotransmitter maps. In keeping with what I've been talking about before, that you develop the method and then you develop the atlases and maps that make use of those methods, Bratislav's group went on to produce the NeuroMaps toolbox. And this is a framework for assembling various kinds of data, whether it's microstructure, function, electrophysiology, receptor data, and allows us to transform between different major coordinate systems, whether it's the CIVET, FSLR, FS average. This is often a fundamental limiting factor, and NeuroMaps allows us to move conveniently and easily through those different coordinate systems. Justine has also gone on to build a brainstem and cortical function architecture with 58 brainstem nuclei in 400 cortical regions. And Justine won the OHBM Carl Zillis Award in Integrative Neuroscience. I'm trying my best not to steal the thunder of the various speakers here. Navona will be talking later about the classroom. Bratislav will be talking later on in this meeting about all the work that he's been doing, so I'm only showing you a very small sliver of the activities that Bratislav's group have been going on. Lindsay Lewis, who's in the audience, Lindsay has spent a long time working on fundamental mapping between spaces, and this is an important one to allow us to map properly between standardized MRI-based atlas spaces like the HCP's FSLR space and the big brain space. You'll see, if you look, that the FSLR brain map, surface map, is very smooth. Obviously, it's averaged across many hundreds of subjects. But the big brain has much more detail in it, and that makes it sometimes difficult to map between these spaces. What you see there on the graph in the middle is the original mapping between these two spaces as derived from the multi-scale surface matching algorithm from David Van Essen, and various iterations that Lindsay has added on to improve slowly the variance across corresponding landmarks. So there's greater and greater correspondence between the big brain atlas space and the MRI surface atlases. Moving on, Anna's lady, Gonzalez Mitjans, did her PhD with Pedro Valdez Sosa and is now in my lab, has been making use of the big brain environment to inform I just let this video rotate a second. Annie has been taking this information to build a high-dimensional neuromass model. Particularly important here is the incorporation of distributed delay connectome tensors in this. Most virtual modeling frameworks do not incorporate temporal differences across individual brain regions, and this is important when you're looking at the sequence of events at electrophysiological timescales. Annie has been working with Alejandro Salinas Medina, who's also in the audience. Alejandro has a talk later on where he's built a generalized framework for simulating distributed dynamical systems, and this has allowed us to incorporate Annie's mathematical modeling into this computational framework. Alejandro was talking on Wednesday, and he is the implementation of the distributed delay neural mass model within that dynamical framework. Annie and Alejandro are working right now in Marseille with Victor Yeza and the virtual brain. There's a lot of complementarity between what we have been developing in the EEG side of the equation with the virtual brain. We'll hear more about that from Annie and Alejandro later on. So this just allows me to come to a conclusion. We've had now 9 separate big brain HIBALL annual conferences in increasingly exotic places. I like that. But it's been always a joy because the scientific content in these meetings is quite extraordinary and exhausting. That's because there are an awful lot of people in HIBALL. The growing community globally. And at this point, depending on how you count it, they're approaching 900 individual researchers in 45 countries who are HIBALL adjacent or HIBALL direct. And I think this speaks to the success of the HIBALL initiative. And I'm sure that Katrin and I will have much to discuss at this meeting about how we take this whole thing forward for the next 10 years. Thank you very much.